Yes, uh, you can start now.
Sure, sir. If you make me also the co-host, later I can admit the participants, sir.
Yes, I have made already.
Okay, okay, sir. Good afternoon to one and all present for today's session. It's a proud privilege to have one of our ex-students from Deshmukh sir side for today's session, where she is going to guide us about facilities available at her place. So let me just introduce her to all the members for today's session. Today we are going to have a session on facilities available at CCMP, and we are having today's guest as Dr. Sheetal Kalme. She is Senior Business Development and Project Manager from Center for Cellular and Molecular Platforms, Bengaluru. Sheetal madam has completed her academics from Shivaji University, and she is having her doctorate degree from Dr. Govindwar sir. A very proud moment for us, where our respected guest speaker has published many papers in national and international publications and journals. So today we are going to have a very wonderful session where we'll get to know the facilities that are available at her organizations and how to approach for the following. So over to you, Sheetal ma'am. Hello. Sheetal ma'am, you can take over the session.
Okay, thank you, Megha, and thank you to Dr. Deshmukh sir for giving this opportunity to share with you what all we do at CCAMP. And I would request if I have an permission to share my screen.
Sure, ma'am, sure.
Okay, just give me a second, I'll share my screen. Yeah, can you see my screen here?
Yes, ma'am, it's visible.
Okay, so I'm Sheetal, and I'm happy to talk to you today, and thank you for taking out your Sunday time to listen to me. So CCAMP is Center for Cellular and Molecular Platform. We are based in Bangalore, and CCAMP is a part of Bangalore Life Science Cluster, where we have 3 other institutes, that is NCBS, National Center for Biological Sciences, which most of you know been a premier life science institute for research. Then we have INSTEM for stem cell research and regenerative medicine, and we have a third institute, TIGS, that is Tata Institute of Societal Genomics, and CCAMP. So all the 3 institutes are morely towards the research in life sciences and genetics and stem cells, while CCAMP has 2 different wings. I'll just give you a brief about CCAMP and then come to the services side. So CCAMP has 2 wings. The one is services, and another one is a bioincubation. In case of bioincubation, we act as an incubation center for startups related to oil biology, depending whether you're working in silico or in wet lab, and depending on your interest, you can just come up with your idea and your glassware and chemicals to our institute, and you can start working as a startup company. We are partnered with different government fundings for startups. Also, CCAMP has its individual funding as well for later stage of startups in biology. I will give you a few details later on at the end of my session. But I will focus currently on services present at CCAMP. We have more than 12 technology platform services available at CCAMP. Please feel free to stop me anywhere if you want to have questions or anything. I will let you know how you can send the samples and where to communicate with us for this. So you can see on my screen that we have more than 12 technology platform services available as the services offered to all kind of research. Where don't worry about that I'm working on plants, I'm working on animals, whether these samples can be submitted. Doesn't matter where the samples are coming from. What matters is the result and the kind of results you're looking for and kind of technology you're looking for. Suppose you have your samples and you're looking for a specific kind of results and you don't know how to achieve it, just give us a call or an email. We'll help you find out which of these technologies will be helpful for you to get the desired result. So we have flow cytometry for cell sorting and analysis, screening with high throughput screening, microscopy with light microscope, confocal, and electron. I will give you a brief of each of these facilities available with us. Genomics, mass spectrometry, structural analysis, transgenic animal, micro devices. So we'll start with mass spectrometry. So we have in total of 12 mass specs available at our institute. All of them are HRMS. We have like high resolution instrument for discovery and quantitative analysis. So we have all the three makes of mass spec from thermo LTQ. So we have triple TOF, Q-TOF machines as well. We have SAIX 7600, thermo fusion, thermo Q exactive, SAIX 5600, then TSQ Vantage for mostly for we used mostly for targeted analysis, SAIX 5500, SAIX 6500, and we also have GC with triple and single cord from Agilent. And we have two new instruments from Agilent, that's 6495 and 6545 is more towards 21 CFR Chapter 11 compliance. So what are the services we offer from mass spectrometry? So as you know, it can have a lot of applications. So we have divided into 4 major divisions. That is proteomics, where the starting material, I'm just saying it in a basic way so that people who are here, who are in graduation would also understand. Pardon me for my basic talks here. So proteomics is study of proteins, where it doesn't matter your proteins is coming from. Once you have a protein precipitate, we can give you a protein ID, then label protein quantification by ITRAC, TMT, and SILAC for peptide and protein quantification, label-free analysis with SWAT analysis or MS1 quantification. Please stop me, or if you have any questions, please let me know. In case of protein ID, where you have a protein and you want to know what protein it is, and we just do the trypsin division, run it on the machine and blast it against the database and let you know what is the protein matching to. In SILAC or TMT or SWAT analysis or label-free analysis, where you want to compare between a test group and a control group and you want to know up and down regulation of protein or even quantify a specific protein there. In case of metabolomics, we can do it in 2 ways. Untargeted metabolomics, where you have a sample, suppose a plant sample, an extract, and you want to know what are the metabolites present in them. So you just do it untargeted analysis where you come to know what are the compounds present in. Second one is targeted analysis, where you are looking for a particular compound to do the analysis there and quantify it statistically. So in that case, we have already developed method on HRMS for these of the list, where we have plant growth hormones, phenolic compounds, amino acid, flavonoids, neurotransmitters, and antibiotics. We also have a list of pesticides currently being developed where we can quantify and identify those pesticides in your sample. So in this case, suppose what if you have a compound which there is no method with us available, not to worry about it. You just let us know the compound and we can develop a method for that specific compound and quantify it for you. Okay, then coming to lipidomics. We do targeted and untargeted analysis for lipidomics. Here again, you will have to, in all these cases, you will have to extract these samples and give it to us. In case of protein, you have to precipitate the protein and give it to us. In metabolites, you have to extract the metabolites. And in case of lipids, you have to extract the lipids and send it to us. Then coming to biologics or biosimilars, mostly concentrating on high molecular weight proteins or bulky proteins, where we form intact mass analysis, reduce, non-reduce analysis, peptide mapping, post-translation modification like phosphonylation, ubiquitinylation, glycomics, those kind of different post-translation modifications, that is PTMs, which can be identified. Disulfide linkage analysis, N-glycan analysis, host cell protein contamination analysis, SECMAS sequence variance analysis, SECMAS for aggregation of your protein, or you have isomers and you want to find out how they are aggregating and what are the molecular masses there. Then CD, DLS, like dynamic light scattering analysis. And these are a few of them which we perform. Just to let you know, we do not do analysis of amino acid sequence by Edman degradation. We do it through LCMS only. And currently, we are not offering N-terminal and C-terminal analysis, but we do offer de novo analysis of your sample. Coming towards genomics, we have platforms from Illumina. We have NovaSeq 6000 for higher molecular weight protein genes analysis, like whole genome sequencing, whole exome sequencing, eukaryotic and prokaryotic analysis, mRNA or small RNA sequencing, or chip sequencing. Where in MySeq platform, which is mostly towards smaller genome like viruses and bacteria and plasmids, again, we offer small genome sequencing, RNA sequencing, target to X16 as metagenome sequencing, and chip sequencing. In this case of in this facility, we will need you to extract DNA, RNA, send us a purified DNA, RNA. We will give you back the FASTA-Q format. Okay, we do not offer bioinformatics analysis because we are more into student catering analysis. So we believe that students should know the basics, and you will offer as a FASTA-Q format. If you still need the bioinformatics analysis, we can connect you with our startups who are, we know, well and good in bioinformatics analysis. You can discuss with them, few of them, and decide which one to go for. In case of mass spectrometry, you have to give us proteins or metabolites or lipids. We'll do the analysis and share you the data, analyze data along with the raw data as well. So in some of the publications, journal ask you to submit the raw data as well, because if sometimes it's a novel data, you have to even submit it to the databases. Okay, coming towards again in genomics for Sanger sequencing, where we offer Sanger sequencing for plasmid PCR products and genotyping as well. Another things which are possible at our NGS facility is single cell genomics, where you can extract RNA, DNA from a single cell and then do the analysis, where we will need a cell suspension or nuclear suspension from your side, where we put it in 10x platform, which segregates the cells, extract the DNA or RNA, where further it has to be put in on NovaSeq 6000, and we can provide you the raw data or FASTA-Q. Other one is droplet digital PCR. This is like a qPCR where droplet are performed and where you don't need a housekeeping gene in this case. So if you're trained in this, you can come and use for per hour basis this machine. Coming towards NMR, where we have two different NMRs, 600 and 800 MZ NMR, Bruker's, but we only have a liquid state NMR, not the solid state. We have 4 radio frequency channels and variable temperature ranging from -40 to 80 degrees Celsius. Coming towards the cryo-electron microscopy, I think currently in India, we were the first to have this National Cryo-Electron Facility, and I think we have another one now in India with this Titan Cryos 300 kV machine. So this we offer services for single particle analysis, cryotomography, micro-OED. Some people who are not in imaging, I just tell you that previously when you purified a protein, you have to crystallize it to get its structure. With cryo-EM, you don't have to crystallize your protein anymore. You can just give the purified 99.8% purified protein to us, and we can determine the structure of it. So these are some of the protein structures resolved by our facility for academia work. This side is all the academia work. We have proteins from 438 kD, which has almost 2.9 angstrom size, to 4.2 as well. So in this case, at least your protein should be 100 kD to go ahead with this analysis. We have also discovered drug discovery projects done with the industries for through this cryo-electron. Coming towards the imaging facility, confocal and super resolution microscopy. Let me tell you, for all the previous facilities, you can send the samples to us by post, by courier, or by cryo PDP or cold chain, depending on the sample temperature required. In this case, you can send us fixed samples on slide. Others facility, you don't have to be here when samples are being analyzed. But in this case, we would request you to come, because you know your samples better and know which part to focus on when we are doing the imaging. So in case of confocal and super resolution microscopy, We have all kind of microscope, literally all kind of, you just name it and we have that microscope. So you just have to write an email that these are the microscope analysis you want to do. And if you are specific towards any microscope, if not, we can let you know, advise you on what type of analysis or what kind of microscope would be suitable for your work. Yeah. Then coming towards imaging facility, again, this is electron microscopy where we have SEM and TM analysis with cryo and room temperatures as well. We also have micro CT for small structure analysis where we can do only non-moving samples. That is, live imaging is not possible here, but sample right from 7 centimeter can be analyzed here. So we get lots of sample from a bone sample to a Drosophila head. We get a lot of dental samples like tooth implants and all. We also have got snake samples. So you name it, we get different kind of samples. So this is an example of mouse femur, and this is a head of Drosophila. Coming towards flow cytometry, where we have different applications from analysis to sorting, cell cycle, cell signal, polyploidy. Other different applications are available. We have all the three makes, BD and BC, as well as thermo. So if you have a preference, or suppose you have specific importance towards specific company make, we have that flow cytometry with us. Then high throughput and high content imaging. Usually most of people where you have 1,000 samples to screen towards, so people use this kind of analysis where we have screening platforms like liquid handler system from TCAN and Biomech with plate readers, multiplex, bioplex leaders. Excuse me. We also have high throughput imaging platforms like Phoenix high content focal imager, thermocellumic array scan imager as well. So you can send these samples to us and we will do the analysis. Even if you need an imaging only, can also be done. Coming towards transgenic animals and models, wherein this is mostly towards the fly facility, where we do CRISPR modifications, and right from cloning expression, we can give you a live fly, a transgenic fly, which can be shipped to you. We can also ship the larvae as well in this case. Mouse genomics engineering facility, currently we are offering transgenic mouse embryos or mating pair currently. If you want to perform any animal-based experiments, we can connect you with our other institute, that is in stem cell research in STEM. We can give you a contact for them so that they can help you in doing your mouse related studies as well. So another facility is a core equipment facility. This could be more helpful for the people near Bangalore or in Karnataka or say nearby state, because these are the facilities like microbial fermenters, liquid handling systems, ACTA purifier, RT-PCRs, 2D, bioplex, quick stand, these kind of machines where you can come and use it per hour base. You just get your columns and reagent and use the machine per hour base. And we do offer training in these kind of all these services. I'll come back to you at the end, one more slide, and I will tell you what are trainings are we offering here. Okay, so these are the equipments, and even if some of the equipment are not there, like we get studies like somebody wants to use spray dryer and all. So even if the list is not here and you want to know whether that machine is available with us, write an email to us. If it is available with us, we'll help you out. If not, we'll be able to help you connect with the people who have this machine and help you out with that. Yeah. Coming towards a medtech rapid prototyping facility. This is mostly towards the startups who are into developing their own machine, where we have a studio design where people come to us with an idea that we kind of need this kind of machine with this kind of reading pattern or something like that. We design the machine, we do the 3D printing, make the machine and make it viable and test it, device testing as well, which has been made there in our facility, and give you the whole machine. We can do this production till pilot scale. We have class 1000 clean room which can be used to packaging of materials, just be 3D printing, laser cutting, CNC milling, hot embossing, which are few of these instruments usually used towards the machine making or instrument making. Yeah. Coming towards the training, I'll just tell you that we offer services in mass spectrometry, genomics, flow cytometry, and every other trainings we have, where we have very less number of people, say from 10 to 15, where we have given hands-on training. Suppose in case of mass spectrometry, we will have training how to prepare a sample, how to run it on the machine, and how to do the data analysis. So most of these things we do with an industry partner. Supposingly in mass spec, we do it with Agilent or SciEx, or in case of a genomic center, as I told you, we do not have bioinformatics support. So we have a collaboration with National Genomic Center Switzerland, Zurich. So they support us in bioinformatics analysis, where expert from CCAMP comes for wet lab analysis, and expert from Functional Genomic Center Switzerland will come here with the bioinformatics analysis, and they give you training in Bangalore at our facility. So these are some examples of the trainings which we give with an industry partner or a well-known partner. And we are, I can proudly say, we are well-known for our trainings as well as our services, the quality of data we offer. And even if few of these trainings are not available with us, and you have a group of, say, 10 people who are interested in specific training, you can let us know and we can arrange that training. Why I'm saying, you will ask, why not for one person and why on 10? Because see, when we ask a company to get partnered with us for these kind of training, they should be ready to spend their time as well. When you have at least a group of 10 people, it's worth their time. That is why we offer this, at least having 10 people and not more than 20 people in a training, because then it happens that people are just craning their necks to even just have a look at the instruments. We have seen this in other facilities. So we have decided to go for a smaller group for training. And other than that, you can visit our CCAMP website to see what are the upcoming trainings. We have recently just finished metabolomics and proteomics training. The upcoming training in case of bioequipment core, we having a training, I think 22nd to 25th, 4-day training, which is based on in Bangalore, hands-on training. This will be on basic techniques like, say, ELISA, then qPCR, PCRs, and gel run agarosas. These are very basic trainings. Mostly will be focusing on, say, MSc students and PhD students who want to be new in biology or want to learn these techniques. Other techniques, other training, we will prefer students who are in the last semester of their MSc or PhD students, because the other trainings are more advanced and you need to have the basic knowledge to learn these mass spectrometry, genomics, and flow cytometry. That will be helpful for you and you will be understand better. And these trainings are not restricted towards students only. We get professors, early career professors and lecturers. We get industry people or training or scientists, and startups. So all these trainings are open for academia, individual person who work as a consultant. We have opened this for startups and for industry. And you can just go to our CCAMP website, go to training and see what are the upcoming trainings available with us. For any services related or even training related queries, you can write on services@ccamp.res.in. I have my email ID with Megha as well. You can collect it from her. You can follow us on Twitter, Instagram, Facebook. We are everywhere. And you can contact me or any other related things on this number, contact number. Other than, I would also like to tell you that for entrepreneurship, who are interested in starting their own entrepreneur, would like to be an entrepreneur, there is a currently government scheme available, which most of you might know, that a big bioignition grant, where you have, it's a idea to proof of concept study, where you know a basic knowledge, where you have an idea and you know it can be converted into a product, and you have a basic theory or you say a market research telling that this could be a new product which could help the society in biology, of course. and you can apply to it to CCAMP and different partners all over India, and where you can get a grant of up to 50 lakhs. Just remember, this is not a research grant. It is a grant where you have been given to convert your idea into proof of concept, where your idea can be coming out as a product at the end of these analysis. So you can get a grant up to 50 lakhs. There are different other grants which are available with CCAMP. Do visit our CCAMP website and come to know about it. You have questions regarding this, write to me on services, and I will connect you with the people who are be able to help. We have different applications and fundings. Like for agriculture related, we have SIP fellows, where we have different programs going on where an individual can apply, and you get say 5 lakh grant. After the completion of it, you get 1 year, 6 months, where you can go into the society and check what are the problems they are facing. So like suppose currently the call is going on for nutrition, where you go into the society, you are paid for that. Monthly salary is given, you go there and check what are the problems faced by the society. You come up with an idea and you develop a solution for that. And end of that, if you want to form a company, the government will support you with the grants, and you can apply later on with other grants as well to take this forward. We have different entrepreneurship competitions going on through BREC, which is another partnership from CCAMP with government, where we encourage different different students, right from graduation to PhD students who would like to make their career as an entrepreneur, and help you get the training required for it. So this is a BREC competition which happens every year, which prices up to 3 crores in form of different offers and different fundings. So keep an eye on our website and you come to know you can apply for it. Selected participants have a boot camps where you are trained for different business aspects as well. Along with that, we have different divisions, like say tech transfer division, which will help you to do the technology transfer, or say patenting, how to help you in patenting. That facilities are also available with us. And please write to us in anything you required. Visit CCAMP website for many other things which I cannot be able to complete, or I would say cover in this lecture altogether. In case of big grant, which I was talking, 16th August is the last date for application. If you want an outreach or get explanation more about the big grant from our side, please let us know, and we can arrange a Zoom call with your institute or your university, where one of our representatives will give a detailed discussion on how to apply and what are the facilities available with the big grant for you to apply. So do let us know if you want to have that kind of lecture as well. Yeah. And I will stop my lecture here, or say my presentation. If you have questions, please feel free to ask.
Thank you, Dr. Sheetal ma'am. It was really a very wonderful session. Now the participants are free to unmute themselves and ask the questions. There are a few questions in the chat box, Sheetal ma'am. The first question is from Dr. Prakash, sir. Is it possible to crystallize peptide of molecular weight 1.8 kilodalton?
So that's what we don't offer crystallization services. We offer molecular structural analysis, where you don't have to crystallize your protein at all. We can do the structural analysis without crystallization, provided that your protein is more than 100 kilodaltons and a purified protein.
Okay. And another question is from Dr. Prakash sir only. How much is it going to cost to purify bacterial peptide antibiotic and determine sequence at your organization?
So see, when you say only peptide map, say peptide ID or protein ID, in gel, it will cost you around 10,000 plus GST. In purification, depending on what kind of purification it is, like from affinity purification, or say from ion exchange chromatography, that will cost you more than the affinity one. And depending on the required step to purify your protein, say 98%. That will totally depend on the projects or the type of protein, and or say recombinant protein or native protein purification.
Okay, ma'am. Amrish sir, if you can unmute and ask your question. Ma'am, there is one more question from Amrish sir. He's from Dharwad. Do you allow BSc students to visit your campus?
Yes, we do. Do let us know, say 1 month prior before visiting us, because then we can have a slot reserved for you to come and visit our residue. Yes.
And will it be like, ma'am, you will be allowing the students to visit all the departments and just show the working of the instruments?
That will not be possible because these are restricted entries. So we can show you 1 or 2 facilities.
Okay.
Currently available if the say in charge is available at that time when you're visiting, because we get I'll just tell you the reason why we cannot visit all the facility, because it takes very long time to visit each and every facility. Yes. And when you say BSc batch, it is at least 15 to 20 people considering the lowest amount, right? True. And we have restricted access to this machine. You need to have clean environment. So every time you can go 4 to 5, 10 or 10 people maximum in the facility at a time. So it takes a very long time to visit all of them. And again, there's a compliance issue where we have a CDA agreement with industries and people who confidential work is going on that machine. If that sample is going on that time, will not be able to show you the working of the machine. As I told you, we have this basic course, and we are trying to get a basic course where we can have BSc or say degree students who could have an grasp of these techniques. We are working on to it. So even we have courses of image analysis as well, which is open to most of the people. And we have already done this kind of image analysis courses with Singhagad Institute of Technology, where we have a 1 month course, say 2 hours every day for a month, or twice a week or thrice a week, where we do online training of students for image analysis as well. So we are trying to get things like that for graduation students as well. I will let you know once we have this.
Sure, ma'am. One more question from Amrish sir. He is in need of SEM and 16S rRNA sequencing. He just want to know what are the service like details regarding that, the fee structure and the entire
in case of SCM, just put an email to services@ccamp.res.in, because we need to know what the sample and the kind of analysis will be required, what sample is in your stage. So depending on that, we will give you the quotation. In 16S RNA, it would be around ranging from 4,000 to 5,000 a sample. Okay. So you can write to services and I will get back to you with it.
Sure, ma'am. One more question from Dr. Prakash sir. Do you analyze residual antibiotics from food sources?
Yes. So we do have developed some methods for antibiotics, not all of them. So let us know what is the antibiotic you're looking for. You just write an email and we can catch up later in detail about your analysis.
Sure, ma'am. With your permission, I would like to share your mail ID as well as official mail ID in the group later. Yeah, so that any queries regarding the services, you can just have mails from the respective participants, ma'am.
Sure.
Yes. Any questions? Any more questions from the participant side? You are free to unmute yourself and ask. Since this is a very interesting session where we have got a lot of information regarding the services available. Any more questions? I guess there are no more questions. So let me end the session here by saying we are really very thankful to you, Dr. Sheetal ma'am, for providing your valuable time on Sunday and giving us knowledge about your campus, what are the facilities available, because it's being an indispensable part of a research scholar, because we have many queries regarding where is 16S rRNA done, where is SEM done, where is STEM done, and it's really not possible for every scholar to forward the samples and get the details done. So just because of what information you have given today, it will be really easy for all our research scholars, students as well, to understand that there is organization like CCAMP, where you can get a lot of provisions done at a time, and even the facilities that you're providing to gain fund from the government authorities, and even encouraging the entrepreneurs for developing their firm. So I'm really grateful to you, Dr. Sheetal ma'am, for giving your time and giving us information about the provisions available at your organization. Thank you.
Thank you, Megha. I just one more question I've seen here for heavy metal. No, we do not do heavy metal analysis. And thank you again to Megha and Dr. Deshmukh sir for giving me this opportunity to talk to you all. Please do let me know if you have any queries on services, or my official ID will be shared by Megha here.
Sure,
Thank
sure,
you again.
sure. I'm also thankful to Dr. A.M. Deshmukh sir for providing us the opportunity to organize sessions like this, where research scholars, even the lecturers, all of us are getting more and more exposure to many stuff that are going around, and we are still blind about that. So thank you, Dr. Deshmukh sir, for giving us this chance of getting the ideas. Ma'am, there is one more question from Swati madam. Do you have any research grants for PhD students?
No, we don't have research grants. As I told you, we have only two wings, for entrepreneurship and for services.
Okay, okay, fine. Thank you. And last but not the least, I would also like to thank all the members today who have joined, taking out time in their busy Sunday schedule. So thank you one and all for today's session, for joining for today's session. Thank you, Dr. Ma'am, Sheetal ma'am. It was really great listening to your information, and we are looking forward to have sessions for Microbiologist Society also, where we can get familiarized with many techniques.
Thank you, Megha. Thank you all for your patience of listening.
Thank you.
Good day.
Thank you. Yes, sir. Deshmukh sir, shall I end the session? Yeah, I guess he is busy. Thank you, ma'am.
Thank you. Good day.
Thank you. Thank you, one and all.